So it gives me great pleasure today to introduce our guest speaker, Dr. Mark Chong from the St. Vincent's. Many of you will know Mark. He completed his PhD here with Tom Kay in 2004, I believe. After that, he went to New York to the lab of Dan Lippman, who was I guess a pioneer in the field of molecular immunology. And he undertook a really fantastic postdoctoral position there. Basically, and that's where his interest in small microRNAs or small regulatory RNAs was really peaked. Having come back from the States, he came back to WEHI as a group leader, continuing on his research into T-cell biology and epigenetics and microRNAs, and then subsequently in 2012 established a laboratory at St. Vincent's. During his career, Mark has
use touch tones for navigation. Press 9 for help.
Responsible for several seminal findings, in particular discovery of alternative
use touch tones for navigation. Use touch tones for navigation. Press 9 for help.
Regulatory mechanism. He's responsible for developing a number of mouse models which have been integral to the field, and has also pioneered the use of high-throughput sequencing in order to characterize processing of RNA. So basically, he's in an ideal position to give today's seminar, which is microRNAs and other small regulatory RNAs. So over to you, Mark.
Thanks, Rat. All right, so today, so I've been charged with telling you a little bit about microRNAs, but I've decided to expand to talk a little bit more to cover other regulatory RNAs, some of which are related to microRNAs and others that are not so much. So I'm going to divide the talk into 3 parts. Most of my time I'm going to spend on microRNAs and related pathway RNAi, and I believe you've already had a lecture from Ross Dickens about RNAi, and it's important to discuss these two together because they're essentially the same pathway. The second part of the talk, I'm going to talk about nuclear siRNAs, nuclear short inhibitory RNAs, and the regulation of transcriptional silencing. And then depending on how time goes, I'll touch on a few other regulatory RNAs, small or short RNAs that we don't know much about at this stage, but may have important functions in biology, and we'll see how we go with time, and I'll just introduce those briefly. So let's start with the first topic, microRNAs and RNAi. So I think by now, most people in this audience have at least heard of microRNAs, whether in seminars or in lectures in undergrad. And basically, microRNAs are called microRNAs because they're these small, short RNAs, typically around 22 nucleotides in length. And they regulate the protein-coding messenger RNAs primarily by translational repression, but ultimately degradation of the messenger RNA. And this has been an intense interest over the last decade or so. Now, actually, microRNAs were discovered a long time, back in the early 90s. And they were first discovered, the first microRNA was discovered in a nematode, C. elegans, which is shown here. And it came out of genetic screens for mutations in cell division pathways. The mutants were generated primarily by Bob Horvitz back in the late 70s, early 80s. And a series of these mutants was studied by his subsequent postdocs, Victor Ambrose and Gary Ruvkin. 2 of the mutants that were of particular interest were this called Lin14 and Lin4, Lin4 lineage mutant, and they were tracking the division pattern of this T cell, which is part of gonad, so it's nothing to do with the immune system in mammals. It's just called the T cell. And basically, in wild type animals, in the larval stage, you get this division pattern that's easily tracked by under the microscope. The Lin14 mutant loss of function resulted in this perturbation in this division pattern, whereas the gain in function, so where multiple copies were introduced into the worm, resulted in this recapitulation of this pattern up here. At the same time, they had this Lin4 loss of function mutant that exactly had the same pattern of division reiteration as the Lin14 gain of function. So Lin14 upregulation was the same as Lin4 downregulation. And basically, it turned out that Lin4 was regulating Lin14, and this work out of both Gary Ruvkin and Victor Ambrose's lab. And by mapping the Lin4 gene, they determined that it was a very tiny region of the gene that was regulating, that was actually functional in suppressing Lin14. It turned out that that tiny little fragment encoded a very small RNA sequence and a slightly longer, what looked to be a precursor sequence. These were, Lin4 was originally termed a short temporal RNA because it regulated timing of cell division, and these sequences turned out to be shown here. So this longer sequence corresponded to this, what looked like a stem loop structure, and the mature sequence turned out to be this short temporal RNA highlighted in red. And both labs showed that this Lin4 basically bound to the 3' untranslated region of Lin14 and regulated the expression of Lin14. So it suppressed the translation of this messenger RNA. And you'll notice that there are multiple binding sites, but none of the binding is perfect. You see all these bulges, and these mismatches are very important in the way microRNAs function. So a few years later, in addition to Lin4, another short temporal RNA was identified, Let7, and it also regulated the division pattern of cells during the larval development. In this case, Let7 regulated another lineage gene called Lin41. So this interplay between Lin4, Lin14, Let7, Lin41 was very important in controlling cell division in the larval stages. And they were, like I said, they were termed small temporal RNAs because they regulated the timing at which the cells divided in the C. elegans larval stages. So for a long time, these two small RNAs were thought to be curiosities of the worm, and for many years, the two labs were studying this, and they were very interesting biology, but no one else really thought very much of it. However, back in the late 90s and then in the early 2000s, a whole slew of papers came out demonstrating the presence of these small RNAs in virtually all multicellular organisms. They were now termed MicroRNAs because most of these small RNAs were no longer regulating temporal cell division patterns, but could have functions, all kinds of biology. So there were many more were discovered in the worm, microRNAs were discovered in plants, in mouse and humans, and many other species. So microRNAs, the microRNA field exploded at this stage, and at this stage we knew that they were present, but not very much about function other than those two small microRNAs, small temporal RNAs. So it's important I bring into, at this stage, the phenomenon of RNA interference. And this discovery of RNA interference occurred around the same time as the discovery of microRNAs. And it was actually discovered in several organisms. This is the best known paper. This was the discovery of RNA interference in animals, work done by Andy Fye and Craig Mello. And this publication led to them winning the Nobel Prize for Medicine in 2006. So in animals, it was coined RNA interference, and what they showed was using a model transgene in C. elegans, which expressed GFP, so the cells glowed green here. If they introduced into the worms long double-stranded RNAs against GFP, you get loss of the GFP expression. Whereas if you introduce irrelevant double-stranded RNA, you don't get loss of GFP expression. So the sequence of the GFP in the double-stranded form was sufficient to knock down the expression of a target gene. And they later showed that it was involved the production of small interfering RNAs from this double-stranded RNA. And I'll come back to this processing in a moment. Now, animals, although RNA interference in animals was probably the best known and certainly best studied, it was not the only phenomenon discovered of RNA silencing. So around the same time in plants, phenomenon of gene silencing was discovered. This was done by several labs, including by Peter Waterhouse, who was at the CSIRO at the time, and they showed basically the same phenomenon where you introduce a double-stranded RNA against a gene, and the introduction of double-stranded RNA knocks down the expression of that RNA. And also another phenomenon of quelling, which is a Neurospora, a fungus, also showed the same effect. Neurospora they express these enzymes that produce carotenoids that causes the fungus to develop this orange color. And when you introduce in double-stranded RNAs against the enzymes that catalyze the production of these carotenoids, you see progressive loss of this orange pigment. So you basically had these various RNA silencing elements, phenomena in animals, plants, and in fungus. So in all clades of all kingdoms of life. So it turns out that RNA interference is dependent on this enzyme DICER. Again, you probably would have had an introduction of this from Ross Dickens. This discovery was from Greg Hannon and Brenda Bay's labs, early 2000s. And by this point, we knew that RNA interference was associated with the production of these small, short inhibitory RNAs, which are shown here in gel. So this is a marker here on the left. So they showed that if you take lysates from here, S2 cells, which are Drosophila cell line, or lysates from Drosophila embryos, and mixed it with double-stranded RNA, you get the production of these 22 nucleotide approximate short inhibitory RNAs. So these are the inhibitory RNAs derived from the double-stranded RNA that go on to silence your target gene. So they surmised that this processing required some kind of double-stranded RNA processing enzyme. And at the time, there were a number of proteins that looked like they could do this job. One was DICER, which had this, which I'll discuss a little bit throughout in the next few slides. Drosha was another one, and another protein that looked a bit like DICER called homeless. Now, the top two enzymes are particularly important because they have these domains called RNase3 domains, which are predicted to cleave double-stranded RNA, and they also have these double-stranded RNA binding motifs, which are predicted to bind double-stranded RNA. So when they immunoprecipitated these proteins and they mixed them with these double-stranded RNAs, and you show that it was DICER that was the one producing these short inhibitory RNAs from the double-stranded RNA. And they go on to show that it is DICER that's causing knockdown in experimental RNAi. In this experiment, they're looking at cells that express GFP, and they've expressed either double-stranded RNA against an irrelevant gene, in this case luciferase, or against GFP. And if you express it against GFP, you get knockdown of the GFP corresponding to the sequence. And now if you interfere with the expression of DICER, you kind of abrogate part of this suppression. So it showed that it was DICER that was responsible for RNA interference. So this is just shown in the cartoon. This double-stranded RNA that you introduce into a cell, that's processed by DICER into these short, approximately 22 nucleotide RNAs, and these short inhibitory RNAs then bind to your target messenger RNA, resulting in the cleavage of that messenger RNA. In this case, just note that we're using siRNAs that are perfectly complementary to the messenger RNA. So within about 6 months of that discovery of DICER being involved in RNAi, it was also determined that DICER was important for the production of those small temporal RNAs that I talked about a few minutes ago in C. elegans, so Lin4, Let7, and several labs showed at the same time. So what it looked like was happening, that there was a microRNA precursor, some kind of short stem loop structure here, that was cleaved by DICER to release this duplex that is kind of like the siRNA. What DICER was doing in this case was to clip off this loop structure, and then one of these strands would then become the effector microRNA. But unlike with siRNAs from RNAi, these microRNAs do not cause degradation, do not cause cleavage of the target messenger RNA. Instead, we get translational repression. So what we appeared to have is two different pathways that now are converging. So on one side, we've got the microRNA pathway that is dependent on DICER, that leads to microRNAs that cause translational repression. And on this side, we have RNAi technology, double-stranded RNAs, also processed by DICER, that led to messenger RNA cleavage. So they almost look like identical pathways, and yet you got quite different outcomes. And what it turned out was this pathway was essentially just a subset of this pathway that's going on. And RNAi is essentially commandeering part of the microRNA pathway. So just let's touch on why we're getting messenger RNA cleavage on the siRNA side, the RNAi pathway, and why we get translational repression from microRNAs. So the effector complex of microRNAs and siRNAs is known as the RISC complex, the RNA-induced silencing complex for RISC. And essentially the core of this complex consists of a protein component and an RNA component. So the RNA component would be your guide RNA would be like your microRNA or your siRNA, and the protein component is an Argonaut protein. And shown here is this beautiful crystal structure of an Argonaut from Dinshaw Patel's lab at Sloan Kettering. This is the protein on this side folded over, and inside you can see crystallized, this is the target RNA complex with, in this case, an siRNA. And this is essentially the effector complex that is used by microRNAs and siRNAs. So a RISC complex is an Argonaut protein plus a microRNA or an siRNA plus other cofactors, which we won't discuss. So why are we getting cleavage on one side and then translational repression on the other side? So the Argonaut family of proteins is actually quite a large family. Shown here are many of the proteins that are found. They're essentially divided into two clades, a larger Argonaut clade up here, and a clade known as Pee-wee proteins, which are similar but functionally quite distinct from these Argonaut proteins up here. Now, if we look up the top here, these are the 4 Argonauts that are found in mammals. In this case, these are the human forms. And it turns out that they're very similar to each other, Argonaut 1, 2, 3, 4. The only difference is Argonaut 2 here has a Pee-wee domain that appears to have maintained catalytic function. So it actually has an enzymatic activity. It's able to cleave some kind of target substrate, whereas the others have not. And in the case of microRNAs, as I mentioned before, microRNAs, as with the Lydin-4 example, largely targets messenger RNAs through incomplete base pairing. So there's a lot of mismatching, and most of the base pairing occurs predominantly at the 5' end of the microRNA, known as the seed sequence. And this leads to basic translational repression and degradation of the messenger RNA. So if this is normal translation going on, a microRNA can affect translation at multiple steps. It can prevent the recruitment of the initiation factors that bring the ribosome. It can interfere with the ribosomes. It can cause the ribosomes to stall, or it can cause the ribosomes to fall off. And ultimately, if there's strong regulation at this level, the microRNA risk complex can cause decapping of the messenger RNA or deadenylation of messenger RNA, which ultimately leads to degradation of the messenger RNA. So you get translational repression and degradation. Whereas siRNAs, as I mentioned, we usually use siRNAs where they're perfectly complementary to the target sequence, and this leads to an actual cleavage of the messenger RNA precisely in the middle of where the microRNA, of the siRNA binding site. The reason why this is occurring is siRNAs, when we use it in an experimental setting, is simply taking advantage of the enzymatic activity of Argonaut 2 specifically. So they're perfectly matched, and when Argonaut 2 is loaded with a small RNA that is perfectly matched to its target RNA, the catalytic activity of Argonaut 2 is cleaving the target messenger RNA. So Argonaut 2 has maintained this activity for other reasons, not to do with messenger RNA degradation. However, we've managed to commandeer this activity of Argonaut 2 to target messenger RNAs for knockdown. So what I've told you now is that microRNAs and RNA pathways are actually the same, and RNA technology simply commandeers part of the pathway, in this case the later stages, specifically DICER and Argonaut 2. So I've already told you, so let's go back to the microRNA component. So I've already told you that DICER is important for the biogenesis of microRNAs. So at this point, so this was about 10 years ago, we knew that DICER was processing a precursor that was from this kind of stem loop structure to produce that microRNA that caused translational repression. Where was this stem loop coming from? And it turns out that microRNAs are derived from long precursor transcripts. Most microRNAs are derived from these independent transcriptional units where the microRNA is encoded by its own microRNA gene. And around 40% of microRNAs are actually derived from intronic sequences of host genes, mostly protein coding genes. So in this case, splicing between these 2 exons which would release this intron and the microRNA that's embedded within the intron. And in this case, the transcription of these intronic microRNAs are dependent on the transcription of the host gene itself. In this case here, the microRNA transcript is dependent on regulation of its own genes. Now, just of note, on average, one of these precursor strands has on average 2 hairpins, so potentially 2 different microRNAs, or if microRNAs are derived from both strands, potentially 4 microRNAs. But it can vary tremendously, so you can have only 1 microRNA stem loop within a transcript, or you can have even dozens of microRNAs with a stem loop. So it turns out that microRNA genes can these particular independent microRNA genes can vary quite dramatically in sizes. And this was work done by us several years ago. First, this is an example of a small microRNA gene. Here's an example of a big microRNA gene. These are on the same scale. And what we've done to map the genes is to use chip sequencing for various histone modifications. So in this case, we've chip-seeked trimethylation of lysine 4 on histone 3, which marks active promoters, and trimethylation of lysine 36 on histone 3 for the gene body. And then down here is the RNA itself. And in this case, this is a polycistronic microRNA cluster. There are 6 microRNAs, and this gene is fairly small. It's a simple transcript, simple promoter of about 4.5, 5 kilobases. In contrast, this is a much more complex gene. There are 2 microRNAs in this cluster here at this end, And the RNA, the gene itself, is about 60 kilobases. You see the promoter here. And what you can see is there's lots and lots of gaps in the RNA, which are actually indicative of splicing. So it's quite bizarre. What's such a large gene? But these are not this is not the largest gene that we identified. We did mapping on a bunch of other microRNA genes, and the largest we found was actually this MIR29 gene contains 2 stem loops, so 2 potential microRNAs. It was almost 170 kilobases, which is quite amazing. If you think about it, microRNA is only about 22 nucleotides, so potentially 44 nucleotides worth of functional RNA, and yet you're transcribing something like 170 kilobases of RNA just to get that. Okay, so the way processing occurs is you get transcription or splicing out of a primary microRNA transcript, and this occurs in the nucleus. Drosha then cleaves off these flanking strands as part of a complex that contains other proteins. It releases this stem loop intermediate, which is exported through exportin 5 out to the cytoplasm, and this is now recognized by DICER, which clips off the loop to generate the mature microRNA. So this is the canonical microRNA biogenesis pathway, and the majority of microRNAs follow this pathway. So as I mentioned, both Drosha and DICER are RNase 3 enzymes. This slide I stole off Tim Johansson. Basically, RNase enzymes are split into 3 classes. The RNase 3 enzymes probably evolved from a very simple protein present in prokaryotes. This is E. coli RNase 3, which is where the name of the domain as well as the family is from. It's a very simple protein, contains an RNase 3 cleavage domain, and a double-stranded RNA binding motif. Simple proteins can also be found in simple eukaryotes, but as you go further along in evolution, you get this amazing diversification of domains, much more complex proteins, and the classes divide essentially based on Drosha and DICER proteins. So in animals, you get this development of appearance of Drosha proteins, and in plants and animals and fungi, the appearance of DICER proteins in many of these. So I've just told you that most microRNAs, the biogenesis is dependent on Drosha and DICER, but that's not the case. I'll just show you one example where this isn't the case. The best described non-canonical microRNAs are the miatrons, because they are derived from short introns, hence the miatron. And what's happening in the biogenesis of these microRNAs is that splicing alone is sufficient to release the stem loop structure. So what we have is a very short intronic sequence here that is capable of folding into the stem loop, and splicing alone releases that stem loop, and then this goes on to be processed by DICER to generate the mature microRNA. Now this intronic production of microRNA intermediates is predominantly occurring in lower animals, flies and worms particularly. This is best characterized in these 2 organisms. And that's because in flies and worms, introns are typically only around 60 to 80 nucleotides in length. So this length here is around the same length of what a pre-microRNA intermediate should be. In mammals and in other higher organisms, it's been very difficult to clearly identify miatrons. Although there's been some description and some evidence that they do occur, it's been very difficult to find these. And that's because mammalian introns typically are hundreds to tens of thousands of nucleotides in length. So even if you get splicing, you still have a large amount of intronic RNA that has to be processed. And in the case of these, processing is dependent on Drosha still, as if those independent transcriptional RNAs. In DICER, there is also no Drosha protein, so Drosha is specific to animals. In plants, the initial processing, the first step is dependent on DICER as well as the latter stages. And that's because the microRNA precursors in plants have a much longer stem structure where there's a lot of base pairing occurring all the way down the stem. And that is sufficient for DICER to clip both ends of the precursor to produce the microRNA in the end. All right, so just to summarize this part about microRNAs and RNAi. MicroRNAs are small regulatory RNAs that target protein-coding messenger RNAs for translation repression primarily, but ultimately this leads to degradation. MicroRNA biogenesis mostly depends on the 2 RNase 3 enzymes, Drosha and DICER. However, there are a few examples where one or either enzyme is not required, and I didn't go to all of these, I only showed you one of the examples. We also know that experimental siRNAs or RNAi actually commandeers and utilizes this microRNA pathway to cleave target messenger RNAs. And this is kind of a byproduct phenomenon, but it's turned out to be a very useful phenomenon for us in biology. So this is all very nice biochemistry. Question is, does it have any importance in vivo? Does it have any function? And one of the things we decided to do, when I started my postdoc many years ago, was to knock out some of these genes to determine if there were any functions. So what we did was to knock out Drosera and Dicer, the two key enzymes, and we did the knockouts in mice. A number of groups also did Dicer in mice, and there were also various mutants in Drosophila and C. elegans. And what we've done here is to cross two heterozygous animals, so male and female, and track the disappearance of the homozygous knockout. So in a heterozygous by heterozygous cross, you expect the Mendelian frequencies of about 25% knockout, 25% wild type, 50% heterozygotes. And if you start very early on in embryogenesis, say at E3.5, so this is the blastocyst stage, you still see roughly 25% of those two, the heterozygous and the wild type, and 50% of the 25% of the homozygous knockout and the wild type, and 50% of the heterozygotes, and same in Drosera. But as you move along in embryonic development, around 7.5 you start to lose the knockout embryos, and by 10.5 you've completely lost either both the Drosera knockouts and Dicer knockouts. So lethal very early on in embryogenesis. So clearly important in embryogenesis, this pathway. We've been very interested in the role of these proteins and the pathways in the immune system, in particular in T cells. For example, I'll show you here, we've been studying the role of this pathway in FOXP3 regulatory cells. So these cells are critical for maintaining immune homeostasis, and we've done this using the Cre-Lox-P system in mice. So we've taken a Cre knocked into a FOXP3 locus and crossed them to flox alleles of Drosera and Dicer that we generated. And basically, it also shows that it's very critical for these cells. Mice in which just the Tregs lack Drosera or Dicer die around weaning age from this systemic inflammatory disease that affects many organs. Shown here, for example, is the lung and liver. So you see lots of inflammation in the lung here, lots of inflammation in the liver. The mice all die from this huge cytokine storm and all this damage going on. Interestingly, in the mice, the regulatory cells are still present, albeit at slightly reduced numbers compared to the controls. And what's happening is these cells, even though they're present, they're completely non-functional. So it tells us the microRNA pathway is very important for the function of these cells. And many labs have gone on to use these mice and other mice to demonstrate the importance of this pathway in a variety of organs and tissues and in differentiation settings. So very important in biology. All right, so that's the microRNA RNA pathway. I'd like to turn to a different topic now and talk about nuclear siRNAs. So nuclear short inhibitory RNAs that regulate transcription, not translation, as in the case of microRNAs and RNAi. So I already mentioned that Dicer and Dicer-like proteins are found in pretty much all eukaryotes. So there are multiple Dicer proteins in Arabidopsis, in rice, lots in animals. Most animals have only one protein, the exception is the Drosophila, which has two proteins. And even in yeast, in Pombi, there is a Dicer protein. So this suggests that at least some of the RNAi components evolved very early on in evolution, even in these simple eukaryotes. And it turns out that the RNAi pathway in yeast is involved in transcriptional silencing, in the initiation and formation of heterochromatin, so silent DNA. Pombi is a very well-characterized organism. It's taught us an enormous amount about transcription, epigenetics, secretion, and so forth. In the case of chromatin, most of the early work on the understanding of chromatin structures was developed in the yeast. So there are a number of very well-characterized loci in the yeast genome, one of which is the pericentromeric regions of the chromosome. These are normally maintained as in heterochromatic states, so permanently silenced loci. And this is important for preventing transcription of anything that's around the pericentromeric regions, and also very important for higher organization of the chromosome. And it turned out that the silencing of this pericentromeric regions is dependent on the RNAi machinery. And that's shown here in this seminal paper from Dinesh Mazad about 10 years ago. And what his lab did was to generate mutants of Dicer, the Dicer1-like protein, Argonaut 1, which is also present there, and another protein that's important in the pathway, an RNA-dependent polymerase, RNA-dependent RNA polymerase here. And the mutants of these resulted in de-repression of the pericentromeric regions. So normally around the centromere, there's very little active histone marks, in this case, trimethylation or lysine 4, whereas there's lots of silencing marks, methylation of lysine 9. And when you have mutations in these RNAi components, you get de-repression, so now active marks appearing and silencing marks disappearing. And they subsequently showed that the silencing of these pericentromeric regions is dependent on this RNA-induced initiation of transcriptional silencing complex, or the RITS complex. And it contains three components: Argonaut 1, this chromodomain protein CHIP1, and an unknown protein called TAS3, and there's nothing like TAS3 that's been identified so far. So we knew at this point that this complex was important. And it turned out that this complex contained small RNAs. And this purification of this complex showed that there was abundant amounts of these, about 22 nucleotide small RNAs. And like small RNAs in high organisms, is dependent on DICER. So it appeared that the RNAi components, siRNAs, was important for this complex. And if you mutate any of the components of the RNAi pathway or this complex, you are unable to recruit this complex to the loci that is supposed to be silenced. That's shown down here. So this is a chip for TAS3, which is part of that complex, at an experimental locus that's supposed to be silenced, and at the centromere here. You see in wild-type cells, there's plenty of the complex there and it's the centromere. And if you mutate Argonaut 1, which is here, or DICER, or that RNA-dependent RNA polymerase, again, you lose that recruitment. So the RNA pathway is essential for recruiting this complex to the loci that's supposed to be silenced. And what's curious, it turns out that the silencing, that pathway was required, was dependent on transcription of the locus itself. So this here is again the part of the pericentromeric regions here, and blown up. And if you mutate components of the RNAi pathway or that RITS complex, you actually get transcription occurring at the locus. So you now de-repress the locus, which was not surprising at the time. But what was surprising was there was de-repression at, I'm not sure how well it comes across, but it's actually transcription occurring in both directions. So if you mutate the components of that RITS complex or the RNAi components or a methyltransferase, you basically get transcription in two directions across the locus. And what it turned out was these transcription across the locus produced double-stranded RNAs, and these double-stranded RNAs were processed by DICER. These are then loaded into that RITS complex containing those three proteins, and that RITS complex then goes back to the locus that's being transcribed to silence that locus and to induce heterochromatin formation. This recruitment of this RITS complex to the locus brings in CLA4, which is a methyltransferase for lysine 9, and the deposition of trimethylation of lysine 9 brings in SY6, which is an HP1 equivalent. Basically, you get this spreading of heterochromatin proteins across the locus, and that's how it's being silenced. So what's going on is you get, for transcriptional silencing to occur in yeast, there's actually a self-enforcing loop requiring transcription and production of siRNAs. So what I'm trying to tell you is you need transcription to silence transcription, which is counterintuitive, but it's really quite an amazing phenomenon. So the other thing it tells us is that these RNAi components actually evolve initially to silence transcription, and translational silencing, in the case of microRNAs, evolved much later in evolution when we started developing multicellular organisms. So for a long time, so one of the obvious questions was, since there's transcriptional silencing going on in yeast, and it's dependent on RNAi components, is this transcriptional silencing by the RNAi machinery still maintained in high organisms, in plants and animals? And for a long time, it was very difficult to determine whether this was occurring. So we knew we had Argonaut proteins. There were plenty of chromodomain proteins in high organisms. The problem was this TAS3. This protein was only found in yeast. Nothing alike, no homolog ortholog could be found in any other organisms. So it was very difficult to determine whether this mechanism of transcriptional silencing was occurring or is occurring in plants or animals. And this was hampered the field for many years. However, it does turn out that RNAi-dependent transcriptional silencing mechanism does occur in high organisms. It's now reasonably well characterized in plants. And in plants, the silencing mechanism follows a similar kind of pattern to what goes on in the yeast. However, the components involved are slightly different. So like in yeast, the locus that's supposed to be silenced is generating double-stranded RNA as a result of transcription in both directions. This is processed by DICER to release small RNAs. These small RNAs are then loaded into a complex containing Argonaut 4 and other proteins that are quite different to what we saw in the Ritz complex in yeast. So the domains of these proteins are not very well understood, and also the function not very understood. However, what it is known is that this now transcriptional silencing complex recruits enzymes that transfer methylation marks onto the DNA into the locus that's supposed to be silenced. So in this case, transcriptional silencing involves DNA methylation rather than modification of histones as occurs in yeast. So essentially, a similar theme that's going on, but the players are slightly different. Now, in animals, it's been much more problematic. We basically have almost no idea, or very little understanding of whether transcriptional silencing occurs in animals. We can demonstrate that RNAi-dependent transcriptional silencing phenomena can occur. For example, this was published 9 years ago, work from Kevin Morris, and they were playing around with these transgenes when they were studying HIV replication cycle. And basically what they found was if you introduce in siRNAs against promoter sequences, you could also knock down expression of the transgene. So if you introduce siRNAs, as in regular RNAi against the GFP, you get good knockdown, as you might expect. But when they introduce siRNAs against what would be a promoter sequence, they also got knocked down here. And if you add in TSA, which interferes with 8 stacks, which is the modification of the chromatin, you have no effect on the silencing, the knockdown with the GFP, because this is occurring in the ribosome, but you do affect the silencing when you target siRNAs against the promoter. So this really suggests that there was something to do with transcription. And when they use nuclear run-on assays, which is an assay for determining whether something is being transcribed or not, you can see a clear effect. So the introduction of siRNA against the promoter knocks down the transcription of this transgene. And we also showed this a number of years ago. This is an experiment that I did myself quite a few years ago. This is now showing that if you introduce in this case, shRNA against an enhancer sequence, you can also knock down expression. This is the CD4 locus. It's got several well-characterized regulatory elements. Important is this proximal enhancer, which is necessary for driving expression in lymphocytes. And when you introduce an shRNA against that proximal enhancer, you get knockdown of that CD4 expression in these cells. So the phenomenon of RNAi-dependent transcriptional silencing in animals does occur. However, we have no idea how it works, what the players are, or whether it's even biologically relevant. So for this part, what I've told you is that in simple eukaryotes, the antecedent RNAi machinery actually functions to silence transcription, not translation. So this is to do with the initiation and propagation of heterochromatin. And therefore, the RNAi machinery actually evolved to silence translation at a much later point. And interestingly, evolved separately in plants and in animals. Curiously, transcription itself is necessary for producing the siRNAs necessary to silence transcription in both plants and in yeast. So we now, because of all the work that's been done initially in yeast and now in plants, we know that siRNA-mediated transcriptional silencing is an important biological mechanism in these organisms. However, in animals, we still know very little, and we're not really sure if it's really important at all, or still, or if there are really any real genes that are actually silencing this mechanism. All right, just in the last few minutes, I'd like to introduce just very briefly a series of other short or small regulatory RNAs. Most of these we know very little about compared to nuclear siRNAs and microRNAs, and these are most certainly going to be turn out to be quite important regulatory RNAs probably in the future as people study these more and more. So one of these are one of the slightly better characterized are known as the piRNAs. These are small RNAs that associate with Pee-wee proteins. Now, Pee-wee proteins are part of the Argonaut family of proteins. They're a separate clade, but they still share similar domain features. But most of them have lost the enzymatic activity of some of the Argonaut proteins. These proteins are not involved in they're not involved in the silencing that we know about, or in translational, the silencing in terms of the microRNA pathway, or in terms of the nuclear siRNA pathway. These piRNAs that are associated with the Pee-wee proteins are different size compared to siRNAs and microRNAs. They're typically 28 nucleotides in length, and they're expressed almost exclusively in germ cells. So they're well characterized in Drosophila oocytes and in mammalian testes. And if you take RNA from either of these and fractionate the RNA, you actually see two equivalent peaks coming out in the cell. The 22 nucleotides, which correspond to microRNAs and other Si Rnas, and 28 nucleotides corresponding to piRNAs. So in germ cells, they're basically as abundant as microRNAs. And they appear to function in controlling genome integrity. So there's now been quite a bit of work done on piRNAs. piRNAs appear to be derived from clusters within the genome. So this is the mapping of piRNAs in Drosophila, and you see that they're located on both sides of the chromosome and in very high density. How they get to this point of a small RNA is still not really clear, but almost certainly involves transcription of these clusters. And these clusters, whether it's a long transcript and processed or transcribed individually, is still a little bit hazy at the moment. But what is known is that these piRNA precursors enter the cytoplasm, where they enter this cycle to produce the effector piRNAs. The cycle known as a ping-pong cycle because it essentially uses the piRNA precursors, the piRNA, and the transcripts from the target sites to form this cycling state to produce more of this piRNA. And this piRNA goes back into the nucleus where it targets transposons for silencing. Transposons are potentially detrimental to the genome because they can move around the genome and cause mutations. And this is thought to be a mechanism of silencing this transposition that's going on. Another example of a short RNA are these promoter-associated short RNAs, or PASs, and they are derived from actively transcribed genes. And if you sequence an actively transcribed gene and just ignore the actual gene transcript itself, what you see is a huge accumulation of these small RNAs in the plus strand and in the minus strand of the gene. So this plus strand corresponds immediately after the promoter, and the antisense RNAs appear to correspond to the opposite direction of the promoter itself. What these small RNAs are doing, we have no idea, but it suggests one possibility is that they are involved in modifying the local organization, the structure of the promoter to regulate transcription. Another example is our enhancer-associated short RNAs, or eRNAs, and they're very similar to those promoter-associated RNAs. Shown here is the binding of RNA polymerase to enhancers. In this case, these are extragenic enhancers. So enhancers come in two general flavors: enhancers that are outside the gene locus, and enhancers that are within the gene itself, frequently in the first intron. So we're ignoring the first intron enhancers in this case. So the ones that are extragenic, you see that there's actually localization of RNA polymerase to the enhancer, and like as the promoter, you actually get transcription across the enhancer. So in the plus direction and on the antisense direction, in the opposite direction, so shown here. So we're getting this kind of double-stranded RNA transcription in both promoters and enhancers. Again, we have no idea what this means. Again, and the postulate is that it's involved in modifying the local structure of the enhancer. And finally, we get these small RNAs that are very similar in size to microRNAs being generated from splice sites. So basically, the 3' end of an exon, and it turns out it is accumulation of RNAs around the 22, 20, 24, so very similar to microRNAs. So when these were identified, there was some thought they might be processed by like microRNAs, but when you knock out the microRNA machinery, in the case DICER or DGCR8, which is part of the Drosha complex, there's no loss of these small RNAs. So they're not part of the microRNA pathway, but again, we have no idea what the function of these things are. All right, so this last part, I've showed you that there are actually many other classes of small or short RNAs in addition to microRNAs and siRNAs, and I think we're probably just scratching the surface at the moment. The one we do know a little bit about is the piRNAs I showed you about, but I've showed you there are lots of RNAs, small RNAs that are generated from regulatory elements, promoters and enhancers, and also from splicing. So I mentioned piRNAs are involved in silencing transposons. Now the function of these other small RNAs is completely unknown and probably involves regulating transcription or local chromatin structure and so forth. And really, it's a lot of work to be done. All right, so just to finish off, I'll show you this. This is the central dogma of molecular biology. Most of us were taught this all the way through high school, through university. So we know that genes, DNA, are transcribed to give you RNA, which the RNA is then translated into protein. So information was always flowing this way. And ultimately, the proteins then function on other proteins and on RNA and DNA to regulate translation, transcription, and so forth. But now we know is that this is way oversimplified. It turns out that RNAs have important functions in their own right. We have lots of RNAs that are functional at the level of DNA, or other RNAs, or on proteins. So in addition to these small RNAs I talked about, microRNAs, siRNAs, we now know that there are long non-coding RNAs that are probably important in regulating transcription and possibly other functions that we don't know about. And of course, I haven't talked about any of the structural RNAs, like small nuclear RNAs, small nucleoled RNAs, and of course, ribosomal RNAs are also functional RNAs. So all of this there are much more levels of regulation, many more functions of RNA than we ever appreciated. And I think we're really just scratching the surface at the moment, and it's really a lot more work to be done. All right, I'll stop there and take any questions if there are any. Thanks.
You didn't talk about this at all, but I'm interested in if you could comment on the challenges associated with high throughput sequencing of microRNAs.
In what sense do you mean the challenges?
Well, okay, specifically the bioinformatics challenge. Because I've got some microRNA sequencing data from the Living High-Seq machine, and I was just wondering, in your experience, if you could comment on sort of the challenges involved from getting your data to actually getting some microRNAs information out of it.
So initially, when microRNA sequencing started, the main challenge was actually trying to capture the microRNAs specifically within a library. I don't know if you've ever encountered that, but that's bypassed by new enzymatic procedures that we can account. The main issue with the main challenge with any kind of sequencing technology is potential biases that occur of the way you construct the library, because you're supposed to you have to capture, in this case, small RNAs with particular chemical modification, and there are inherent biases in that, as well as having PCR in amplifying the library. So that's one aspect that you have to take into account. And high throughput sequencing is excellent for profiling an overall population of RNA, but it tells you the overall population of the microRNA. It doesn't tell you an absolute quantification of microRNAs, for example. So as I alluded to, the absolute quantification issue, because when you do sequencing, you're basically taking everything in there and then sequencing as much as you can from that pool. So you don't know how much it is absolute there, but you do know what the relative amounts of the sequences are within that pool. So ultimately, we do always go back to some more quantitative measures like quantitative RT-PCR. The other issue is mapping the microRNAs. So lots of us in the field have just mapped lots of small sequences and called the microRNAs and deposited into the databases. To be honest, I think at least half of the microRNAs that are in microRNA databases are probably not microRNAs in terms of functional microRNAs. They're there because they've been pulled out in small RNA libraries and they look like a microRNA in terms of the structure sequence. But whether they actually enter the microRNA pathway and regulate translation, that's a whole other matter, and you still need to do the biology to do that. But that's certainly probably the main areas of issues.
I was just wondering where we're at with using siRNA as a therapeutic, and obviously there's problems with delivery of it, but if they're small portions of RNA that you can put into a cell and pretty much control the expression of any protein, I mean, is anyone doing anything trying to do that?
There's a huge industry trying to take RNAi to clinic. So the delivery is obviously one big issue, and there are a lot of people trying to work on that problem. One thing that we've been interested in is the issue of the targeting itself. So an siRNA that you target a gene to, so you have a perfect sequence and you target the gene that you're wanting to knock down perfectly, you get the cleavage reaction, that's well and good. But the problem is it's essentially hijacking a pathway that is recognizing targets by mismatches. So that's where our main issue of what we used to call as off-target effects. But it's not really off-target effects. The machinery is designed to recognize targets imperfectly, at least in animals. So that's where our main issue is, really. So you're probably affecting lots of other potential targets, not just the target that you're affecting by cleavage.
What do you think those microRNA genes that are enormous and then the microRNA itself is tiny? What's the function of the rest of the RNA, or if it's just a funny twist of evolution?
Yeah, that's a good question. So the question is, what's happening to those RNA transcripts? So we've got this huge, long primary microRNA transcripts, and the process down to this very small amount of RNA is actually functional. What's happening to the other RNA? The question is, we don't know why they're being transcribed that way. we actually find it very difficult to. The reason why we can see it in our RNA-seq analysis is we did a little trick. We actually blocked our microRNA processing to be able to detect it in the cell. So in a normal wild-type cell, most of those transcripts are degraded very quickly. So once the microRNA intermediate is excised out of the precursor, the flanking RNAs are degraded almost instantaneously. Only a handful of microRNA precursors are present in the cell in any appreciable amounts. Question is what they're doing, I have no idea. Why they're so long, we don't know. When we look at the obvious things like whether there's conserved sequence conservation across species, we only find very small portions of those flanking RNAs being conserved between other mammals, for example, between mouse and man or other mammals, and only these very small islands of areas that are highly conserved. And we suspect those islands of high conservation are probably regulatory in some way, but the rest of it, we have no idea. It is very curious.
Is there like a positional bias? So you, in your example, you showed that the microRNA was at the very end, because then you could imagine that, for instance, it takes a while to transcribe that one thing. So it would take a while to come to the microRNA.
There is a bit of a mixture in terms of where the actual precursor is sitting within the transcript, but you're right. In a lot of cases, it's towards the 3' end of the transcript. Why you need that long transcription, again, we're not really sure. Like I also mentioned, you see a lot of splicing goes on in these primary transcripts. We don't know why that would go on. I mean, the possibility is you need splicing to get that long transcription to occur, and that's quite well known. Splicing promotes transcription, but why do you need that long transcription? Again, you can only speculate.
What do the promoters look like? Do they look like the normal promoters that you can see of the transcriptions?
So the microRNA genes? Yeah, so microRNA gene promoters look like promoters of any other protein coding gene. In this case, it's essentially a non-coding RNA gene, and it's regulated by transcription factors. We don't know many of those, so BCL6 is one transcription factor that looks like it regulates like near17 cluster, for example. But not many people have done much work on the regulation of microRNA genes, because it's only very, very recently we've been able to visualize the microRNA genes themselves.
So you've shown that in, for example, the T rate reducible knockdown, that the disruption and dice phenotype are pretty similar. But I'm also aware that you've shown that in certain cell types that's not the case and they're not overlapping. Do you think that's a differential role in this transcriptional silencing method?
So so the question, so if you have two enzymes that are important for the same pathway, you knock them both out, you should get the same phenotype. So in the case of the microRNA pathway, since we know that DROSHA and DICER are important for microRNA biogenesis, if you knock both of them out, you get the same phenotype. You can pretty much assume that it's probably all related to that pathway. But as Grant alluded to, we've done lots of knockouts now with a lot of other labs looking at different tissues. We don't always get an overlapping phenotype, identical phenotype. One good example is Tim Johansson, PhD student, who's been working on dendritic cells, and we know that the DROSHA knockout has a severe effect on dendritic cell development, but not DICER knockout. And that's because DROSHA is doing something else in addition to microRNAs. In this case, DROSHA is regulating messenger RNAs directly. DROSHA can actually cleave messenger RNAs, but only in stem cell and progenitor type cells. DICER, on the other hand, has an effect on siRNAs. There are some examples in germ cells where you knock out DICER, you have much more severe phenotype in germ cells compared to the DROSHA knockout, and it's probably related to those endogenous siRNAs. And then we've also had one other example where we knocked out in the eye, comparing DICER and DROSHA, and there's a huge, there's a terrible phenotype in the eye. When we knock out DICER, basically you get this macular degenerative disease in the eye, occurs in mouse and humans. And that's the function for DICER in cleaving RNAs derived from sign elements, and the sign elements are actually toxic. So that's got nothing to do with siRNAs, but nothing with microRNAs together. So it depends on context. There are some examples where DICER has a specific function, DROSHA has a specific function in addition to microRNAs, but in most cases, in most cell types, especially somatic cells, the two enzymes are predominantly functioning in the microRNA pathway.
Very interesting talk, Mark. You didn't talk too much about targets of microRNAs, specific targets. Has there been progress in identifying, since they seem to regulate many different targets, has there been progress in identifying which are the more relevant ones?
Yeah, that is an important question. So how do microRNAs recognize their targets? So as I alluded to, a difficulty in the field has been this, at least for animals, is the recognition of targets is always mismatch. It's almost invariably mismatch. And even the best examples of matching, you still get one or two mismatches occurring between the target and the microRNA. So that has hampered effort in trying to predict targets. How do you predict a target if it's not dependent on sequence and dependent on sequence at the same time? So it's been very difficult. There's been lots of groups trying to develop algorithms to predict targeting based on sequence conservation between species, the presence of motifs, clustering of potential microRNA sites, but none of them look very good. So when you do an experiment where you either knock out the microRNA or you overexpress the microRNA, how well does it correlate with prediction algorithms? Usually not very well, at best 10%, 20%. So there's been other ways trying to address this issue of how to determine targets. One good example is trying to actually capture the complex itself in which the microRNAs are binding to the messenger RNAs. And a lot of work has been going on in recent years where you basically immunoprecipitate the complex and try to sequence everything out so there are things that actually co-immunoprecipitating together and then inferring back what the targets are. Ultimately, you have to go, most people still have to go back and do the experiment where you mutate the site that you think are important or use transgenic methods. So it's still, there are high throughput mechanisms, but you still have to go back to biology just like anything else.